Okay, we'll go ahead and get started. So before or last week after the exam, we finished up gene expression, but we didn't go through the lecture questions, so we're going to start today by going through the lecture questions for gene expression, and then we'll move on to new topics. Okay, so the first question that I want to go through from this lecture questions is the first one, which is what two processes have to occur to produce a protein from a sequence found in DNA? Right, transcription and translation. Right, and in that order too. Number 7, where does transcription begin and end in bacteria? So what I'm asking is, what's the segment of DNA that signals the beginning of transcription, and what's the segment of DNA that signals the end of transcription in bacteria? Right. Okay, so the promoter signals the beginning, and the terminator signals the end. And in eukaryotic cells also have promoters, and that will signal the beginning, but then they just have a different segment of DNA. It's called something else that signals the end of transcription. You don't need to remember it for this class. Okay, and you also remember that the promoter, in terms of being involved in transcription, is what binds the promoter, is what binds RNA polymerase. So RNA polymerase binds at the promoter, and that tells RNA polymerase where to start. Number 13, what does it mean when we describe the genetic code as degenerate?
Multiple nucleotide groups can code for the same codon.
Close. So multiple codons can code for the same amino acid. Yeah. So when we say the genetic code is degenerate, that means that a particular amino acid can be specified by more than one codon. So that's what it means to be degenerate. Okay, so we talked about different types of RNA being involved in translation: mRNA, tRNA, and rRNA. In which type do you find codons? So specifically codons, and anticodons being something different. Right, mRNA. So mRNA is the type that has codons. How many nucleotides make up a codon? Right, 3. Our anticodon, now we're talking about anticodons, what type of RNA has those? Right, tRNA. And how many nucleotides make up an anticodon? Yeah, also 3. And can someone explain the relationship between anticodons and codons? Okay, so you would say a particular anticodon is what to a particular codon? It starts with a C, complementary, right? So an anticodon of the tRNA is going to be complementary to a particular codon in mRNA. Number 18, what type of RNA has an amino acid attached to it? So I gave it a specific name. Oh, sorry, no, I'm not looking for that. In terms of mRNA, tRNA, or rRNA, which type has an amino acid attached to it? Right, tRNA. So there's two things that you should remember about tRNA. So every tRNA molecule is going to have a single anticodon that is complementary to one of the codons. And then at the other end of the tRNA molecule, you're going to have an amino acid attached to it. Number 18, what type of RNA... the second part of that question, what attaches amino acids to these RNA molecules? So we talked about a class of enzymes or type of enzymes, and their job in the cell is to attach a particular amino acid to a particular tRNA molecule. It starts with an A. Yeah, aminoacyl tRNA synthase, not acyl. Yeah. So aminoacyl tRNA synthetases. Okay. Number 24, during the process of, so these questions are kind of tricky because what they do is, these are taken from your textbook, they kind of mix up things that happen during transcription and translation in the answers. So during the process of transcription in a eukaryote, which of the following occurs? Right. Okay, so RNA polymerase synthesizes the nucleotides 5' direction. Just to go through the other ones, remember so in A, ribosomes are involved in translation, and they can be recruited to the ER in eukaryotic cells. B, DNA polymerase is involved in DNA replication. In addition to that, it also does not unwind the DNA duplex during that process. That's the job of DNA helicase. And then D is incorrect because during transcription you're not copying RNA into DNA. And then E, the genes are always going to stay in the nucleus in terms of a eukaryote. You don't ever export the genes out. What gets exported out is the mRNA molecules that you make during transcription. Those will be exported out of the nucleus and into the cytoplasm. Okay, so 25, during the process of translation in a eukaryote, what? Not B. Not D. It's E, right. So let's go through each of them. So the first part, so messenger RNA is spliced in the nucleus, but that is not a part of translation. That's a part of RNA processing. Okay, so A is a part of RNA processing, not a part of translation. B is incorrect because we never talked about how amino acids are synthesized. RNA polymerase is the enzyme that synthesizes RNA during transcription. And then C, transfer RNAs are joined together by a large enzyme complex, so you don't ever join together transfer RNAs during translation. And then D, remember that in a eukaryote, the ribosomes are in the cytosol or cytoplasm. They're outside of the nucleus, and they don't get imported into the nucleus. What happens is the mRNA gets exported out of the nucleus, and then they can interact with ribosomes that are outside of the nucleus. And then E is the correct answer. Okay, so 27 is asking you to fill in the blanks with the word bank given. So the first one, in transcription, the goal is synthesis of what? Right. And then in DNA replication, the goal is the synthesis of, right, DNA. And then RNA polymerase and primase both add nucleotides to a, right, okay, and that would also be true for DNA polymerase, okay. So DNA polymerase, RNA polymerase, and primase all build strands of nucleotides in the 5' to 3' direction. Okay, the next one, during blank, both strands of DNA will function as a template. Right. And then during blank, only one strand of DNA will function as a template. Right, transcription. Okay, so this is gets at a question that we asked during the short answer part, but the genetic code is degenerate. So what does that mean again? No. No. Is B. Right, a particular amino acid can be specified by more than one codon. Okay, moving on. Okay, so this is kind of a table comparing transcription to translation and looking at the enzyme, the template, what tells you where to start and what tells you where to finish. So what's the enzyme in transcription that builds the RNA molecule?
Right. And what's the enzyme in translation that builds the protein? No. So mRNA is not an enzyme. Right, the ribosome. What's the template in transcription? Right, DNA. What's the template in translation? Be more specific. Right, mRNA. In transcription, what tells you where to start? Right, the promoter. And in translation, what tells you where to start? Right, start codon. And then in transcription, what tells you where to stop? So that's the termination. Right, terminator. And then for translation, it's what? Right, stop codon. So just make sure, I think the most difficult part is just keeping those straight, which one goes with transcription, which one goes with translation when you're being asked multiple choice questions. So 35, a difference between bacteria and eukaryotes in translation is what? Mm-mm. It's not A. Right, great. C. So this one was a tricky one. So let's go through all of them again. So A is not right because translation can begin on mRNA still being synthesized in bacteria, not in eukaryotes. So remember, transcription and translation can happen at the same time in bacteria, but they cannot happen at the same time in eukaryotes. And then B, in bacteria, a 5' cap is added to the mRNA before translation. So the 5' cap is something that's added to the mRNA in eukaryotes before translation. And then C, in eukaryotes, modifications to the 5' end of the mRNA help to binding the ribosome. So this was the right answer. And this goes back to when we discussed the 5' cap and the 3', sorry, we didn't call it, and the poly-A tail. And I told you that in terms of their function, the 5' cap and the poly-A tail are important for helping ribosomes bind to the 5' end of the mRNA once it reaches the cytoplasm. D, the genetic code is nearly universal, so bacteria do not use a different genetic code than eukaryotes. And then E, there is no tRNA that reads the stop codon. So does anyone remember what binds to the A side of the ribosome when a stop codon comes in? So it's not a tRNA molecule, it's a protein with a specific name. And it's for bacteria and eukaryotes. Starts with an R. It's 2 words. And the second word starts with an F. No. But those do, yes, that is an R and F. Yeah. Right, release factor. Release factor. Okay. Yep, I have more. 42. After the ribosome moves to the next codon on the mRNA, so now this has to do with translation, the growing polypeptide chain is found in which position? So we talked about the fact that the ribosome has 3 sites that bind tRNA. This is basically asking which site binds the tRNA molecule carrying the growing polypeptide chain. Right, the P site. And does anyone remember what the P stands for? And so it's peptidyl, so the peptidyl site. 43, the codon on tRNA matches up with the complementary anticodon on mRNA. No. Why is that not true? Right. So it should read the anticodon on tRNA matches up with the complementary codon on mRNA, so it switched anticodon with codon. Okay, and then 46, splicing joins together what? Right, B, 2 exons. So remember that what you're going to do in the process of splicing is you're going to cut out the introns and you attach the, or join together the exons. Okay. 50, blank is to transcription as blank are to translation. Right. So it's comparing the two enzymes that are involved in those processes that are involved in synthesizing the product, so RNA polymerase and ribosomes. 53, intervening sequences that are transcribed but not translated into protein are called what? Right, B, the introns. So remember, the introns, when we just define them, they are the sequences that are the non-coding regions that interrupt the coding regions, which are the coding regions being called exons. 54, which of the following would occur if a cell's spliceosomes were mutated so they no longer function normally? Right, A, introns would remain in the mature mRNA. 55, blank enables a single gene to encode for 2 or more polypeptides that are different in their amino acid sequence. Right, D, alternative splicing. So we said that alternative splicing was basically splicing the mRNA molecule in different ways, and when you alternatively splice it, in some cases you can leave out certain exons during that splicing process. And if you splice the mRNA differently, then that's going to lead to a different protein being produced. Okay, do you guys see any other questions from this topic that you want to go through? So this topic has a lot of details to it, in addition to a lot of new vocabulary, so. Okay, so we're going to start a new topic then, which is mutations in DNA repair. And so we're going to start off by just defining what a mutation is. So a mutation is a change in the nucleotide sequence of DNA. Okay, so anytime you change the nucleotide sequence of DNA, that's going to be a mutation. And so because certain segments of DNA that we call genes carry the code, their sequence of nucleotides code for the protein, if you have a mutation in a gene, that can result in changing the amino acid sequence of a protein, and then that in turn can affect the function of a protein. Okay, so we'll talk about mutations within genes, but then we'll also talk about the fact that mutations outside of the coding regions of DNA can also have drastic effects on an organism. And then in terms of what happens because of a mutation, some mutations have no effect whatsoever, and some are going to be minor, and then others are going to be catastrophic. And it depends on a lot of different things. So it depends on the amino acid within the protein that you're changing, how important the protein is to the organism's survival, that you're causing to be different, etc. Okay, so the first thing we're going to go through are different types of mutations. And so basically the first four here are different ways of altering the DNA, the nucleotide sequence. And then the last four, they're defined by what effect they have on the proteins that are produced in terms of the names of those mutations. So the first one is a point mutation. This one's relatively easy. So any mutation that affects a single nucleotide base pair. Okay, so any mutation that affects a single nucleotide base pair is a point mutation.
Okay, so if I have a sequence of nucleotides A, T, G, C, A, C, A, and here's the complementary strand. Okay, so this is the DNA sequence. This is the normal DNA sequence. So let's say that this right here is one nucleotide base pair. And if I were to have a mutation that changed that nucleotide base pair to an AT base pair instead of a GC, that would be a point mutation, okay, because it only affects that single nucleotide base pair, and everything else remains the same. Okay, so if you only affect one nucleotide base pair, then that's going to be a point mutation. And similarly, if I were to just get rid of this GC base pair and only that GC base pair, that's only affecting that nucleotide base pair, and so that would also be a point mutation. If rather than deleting something, I added an extra nucleotide base pair in here, and I only added one, that would also be a point mutation. So as long as you're only affecting one, it's a point mutation. Okay. The next one is also relatively, a given name is relatively easy to remember. So a base substitution is when you take one nucleotide base pair and replace it with a different nucleotide base pair. Okay, so a base substitution is when you take one nucleotide base pair and replace it with another nucleotide base pair. So basically you're taking one nucleotide base pair and substituting another nucleotide base pair for it. Okay, so this point mutation that I drew up here, where I changed the GC to a TA, that's also a base substitution because I substituted a TA base pair for a GC. Okay. Also, if I were to change it, I just want to make sure that everyone's clear on this. If I were to change it to the C to a CG, that would also be a base substitution because this is different from that. Okay, so this would also be a base substitution. Okay, and insertions are also relatively easy to remember. So insertions are when you add one or more nucleotide base pairs. Okay, so insertions are the addition of one or more nucleotide base pairs. And then deletions are the opposite of that. Deletions are the removal of one or more nucleotide base pairs. Okay, so deletions are the removal of one or more nucleotide base pairs. Okay, so point mutations, base substitutions, insertions, and deletions are changing the nucleotide sequence in different ways. And then the last four, their names come from the effect that they have on the protein that's produced by the gene that codes for that protein. Okay, so a frameshift mutation is any mutation that shifts the reading frame. Another way of saying that instead of shifts is it's any mutation that moves the reading frame, and I'll explain that in just a second. So
It's any mutation that moves the reading frame. Okay, so what I'm going to do is this is easier just to draw out the mRNA sequence to explain a frameshift mutation, but remember that the sequence of nucleotides in DNA is determining the sequence of nucleotides in the mRNA molecule. So let's say that this is your mRNA molecule. Okay. And so remember that when we talked about translation, we said that you're going to start at the start codon, so that tells you where to start translating. And then we said that in addition to telling you where to start, it also sets the reading frame. So that means because the start codon is here, then you're just going to read the next 3 nucleotides as the next codon. And then you read the next 3 after that, and then the next 3 after that. So this start codon sets this reading frame. It determines what 3-letter words you're going to read as you move down the mRNA molecule. Okay, and any type of mutation that moves that reading frame is going to cause a frameshift mutation, and frameshift mutations can be caused by insertions and deletions. Okay, so let's say that in the DNA sequence you have an insertion. That insertion is going to show up in the mRNA molecule. So let's say that you have an insertion right here. So there's an insertion in the DNA that codes for this mRNA molecule that adds in an extra, let's say, cytosine. Okay, now you still read AUG as the start codon, and then you read ACG. Now instead of reading ACC, you read CAC, and now you've shifted, you've moved the reading frame. And then rather than reading UCG, the next codon you read here is CUC. So basically, when you have an insertion like that causing a frameshift mutation, all the codons after the insertion are going to be incorrect. Okay, and that's you've shifted the reading frame in that case. And you can do the same thing if you were to delete a nucleotide base pair. So if you delete that, that would also shift the reading frame. Because then what's going to happen is you're going to read ACA as the next codon rather than ACG. Okay, is everyone okay with frameshift mutations? Okay, so silent mutation is a mutation in a gene that does not alter the amino acid sequence of the protein. So a silent mutation is a mutation that occurs within the gene, but it does not change the amino acid sequence of the protein. So can someone explain how silent mutations can take place? So if it's within the coding region of a gene, how can you change the DNA sequence but not change the amino acid sequence? Right, because they're degenerate. The genetic code is degenerate. So if you have a mutation that changes one codon, that specifies, into another codon, and they both specify the same amino acid, then you've changed the DNA sequence, but you're not going to change the amino acid sequence. So basically it's called a silent mutation because the mutation doesn't show up in the protein. So it's considered silent, it's not expressed. Okay, a missense mutation is a base substitution that causes a change in a single amino acid within the amino acid sequence. Okay, so a missense mutation is a base substitution that causes one amino acid to be different in the amino acid sequence. So another way of thinking about this is that there are base substitutions that cause missense mutations. So they're going to cause and that's easy to, I think, visualize. You have a codon, and if you mutate it to a different codon through a base substitution, now it codes for a different amino acid, that's going to lead to a missense mutation. Okay. And then the very last one is nonsense mutation. A nonsense mutation is when you change or have a mutation that changes a normal codon into a stop codon. Okay, so a nonsense mutation is when you have a mutation that changes a normal codon into a stop codon. And so basically, if that happens, then what you're going to do is you're going to stop prematurely making that protein, because once the ribosome reaches that new stop codon, it's going to stop at that stop codon and not continue. And so you're going to produce a shortened version of that protein that's missing some of the amino acids, depending on where the stop codon mutation happens. Do you guys have any questions about the different types of mutations?
So is it not considered nonsense when, like, say the AG got transferred to something like really far?
So, sorry, say that again.
So say like the AG got transferred to something,
What AG? Sorry.
say the start codon.
Oh, AUG.
Yeah, got transferred like somewhere really far down the chain.
Well, you wouldn't transfer. I don't see how 3 nucleotides would be transferred together. If you had a mutation that got rid of the start codon, then you would not know where to start, and you wouldn't even produce the protein. So if you had a point, let's say if you had a missense mutation that changed this AUG to like an AUC, then the ribosome would never make the protein, because there's nowhere telling it where to start. Yeah. Okay, so a missense mutation is a base substitution, so that's what causes it. And it's a base substitution that causes a single amino acid change within the amino acid sequence of the protein. So if you have a mutation that causes one amino acid to be different within the amino acid sequence, that's a missense mutation. Okay. So this is just an example of a base substitution within a gene that codes for a protein that ends up having a drastic effect on the organism. So the mutation that causes sickle cell disease is caused by a base substitution that results in a missense mutation. So you can see that here's the normal gene, or the mRNA from the normal gene. We have a GAG here that codes for glutamine, and then, glutamate, and then this glutamate in people who have sickle cell, they have a mutation that changes this A to a T, and now that codon codes for valine instead of glutamine. And so basically you have this missense mutation that's caused a single amino acid change, and just by changing that a single amino acid, you have sickle cell disease. So that's an example of where a small mutation can have a drastic effect on an organism. And you don't need to know this part that I'm going to tell you, but what happens is when you have this missense mutation, it ends up causing the protein that it codes for to clump together, so the proteins clump together inside the cell. And then that clumping causes the red blood cells to sickle, and it decreases their ability to carry oxygen. And the gene that codes for the beta globin gene is a part of hemoglobin, which carries oxygen in your red blood cells. So again, even small mutations can have a drastic effect, but there are also examples where a mutation will have no effect on an organism. But this is the specific example that you guys went through in lab when you did the, I can't remember the name of it, but one of the labs you did transcription and translation, and you compared the normal beta globin gene to the mutant beta globin gene. Okay, so then you can also have mutations that are outside of the region that actually codes for the protein. So you can have mutations in DNA that does not code for a protein. And they can have an effect in terms of what happens inside the cell and into the organism. So you can have mutations that occur to the promoter. And remember, the promoter is the segment of DNA that binds RNA polymerase. So you could have a mutation, and the mutation may cause an increase in transcription or a decrease in the rate of transcription, and that's going to affect gene expression in the organism that has that particular mutation. When we get to gene regulation, we'll also see that there's segments of DNA that are involved in regulating the expression of genes. Okay, so meaning that what we're going to see is that there's proteins that bind to these regulatory segments and then will maybe either lead to an increase in transcription or a decrease or inhibition of transcription. And so if you have mutations in these regulatory elements, that can alter how the gene is regulated. It could lead to the gene not being expressed, or it could lead to the gene being overexpressed in the organism. You can also have a mutation in the splice junctions or in the segments that tell you where to splice a particular mRNA molecule. And obviously if you don't splice it correctly, that's not going to code for the correct protein. And then there's also these additional ones that can have either an effect or no effect on the organism, but they're both outside of the coding region of DNA. Okay, do you guys have any questions about the effects of mutations that can occur outside of the coding regions? Okay. So the next thing we're going to tackle is just the distinction between mutations in germ cells versus mutations in somatic cells. And just to remind you, germ cells are the cells that give rise to gametes. So just to put that in different terms, germ cells are going to give rise to the cells that carry the DNA that you're going to pass on to offspring. Okay, so germ cells give rise to the cells, those are which are gametes, and the gametes carry the DNA that you're going to give to your offspring. Okay, and then somatic cells are basically every other cell of your body except for germ cells and gametes. Okay, so with the exception of germ cells and gametes, every other cell within your body is a somatic cell. And so the key difference that I want to point out here is that mutations in germ cells can be passed on to offspring. Okay, so if you have a mutation in a germ cell, and then that germ cell produces a gamete that carries that mutation, and then that gamete is involved in a fertilization event that gives rise to your offspring, then basically every cell of your offspring is going to have that mutation in it. Okay, so mutations in germ cells can be passed on to offspring. And then in contrast to that, mutations in somatic cells are not passed on, are never passed on to offspring. Okay, so mutation in a somatic cell will never be passed on to offspring. That does not mean that they're not important, because mutations in somatic cells can lead to things like cancer. It can also cause other problems within the organism that they occur, but they're not going to be passed on to the offspring. There's aspects of cancer that can be hereditary, meaning there's aspects that make it more likely that you're going to get cancer. But cancer is most cancers are caused by mutations to the DNA of somatic cells within the person who gets cancer. Yeah. So in terms of that, just to kind of answer that a little bit more, so remember we said that it takes multiple mutations to go from a normal cell to a cancer cell. And so basically you could, let's say it's 5, it's not 5, but let's say it's 5. So let's say that you inherit 1 mutation from your parents. That means you only have to acquire 4 over your lifetime, whereas another person would have to acquire 5. And it's going to be a lot harder to acquire 5 than it is to acquire 4. And so that's how certain mutations can make it more prone that you're going to get cancer, because you acquire a mutation that is already leading to cancer before you're even born. So, okay. So that's that. Okay. This I'm not going to ask you about in terms of questions on an exam or a quiz, but students often ask these questions, so things that cause changes in the nucleotide sequence. And so you can have, basically they're showing you ones that can arise spontaneously. And so there can be errors in DNA replication that result in mutations. And then there's also obviously things that can cause mutations that are in the environment, things like UV radiation, cigarette smoke, which most people are aware of, and other types of chemical agents that can cause changes to the nucleotide sequence of DNA. Okay, so those are some of the causes of mutations. Now we're going to talk about the fact that cells have the ability to repair DNA damage. And there's multiple pathways for this. The two most common are on the board there, base excision repair. One's called base excision repair, sorry, and the other is called nucleotide excision repair. And really we're only going to discuss the nucleotide excision repair pathway in bacteria, specifically in E. coli, and we'll talk about specifically how that takes place. This is just comparing the two. So on the left you have base excision repair, and on the right you have nucleotide excision repair. So the different enzymes that are going to be involved are obviously going to be different, but then you can also see that in base excision repair you're excising only a single nucleotide, whereas in nucleotide excision repair you're going to excise a segment of a strand of DNA that contains the mutation. Okay, so let's talk about how nucleotide excision repair happens in E. coli. And there's really nothing to write down because everything, this is taken from your textbook, and everything you need to know, the steps are already written on the slide for you. So basically I'm just almost reading from the slide. So what's going to happen in this case is this is a mutation, it's called a thymine dimer. What happens in a thymine dimer is these two thymines that are in the same strand become covalently attached to each other. And that's what a thymine dimer is, and that can lead to a mutation because it can lead to mistakes during DNA replication. So that's what you're trying to fix in this particular example. And in E. coli, the proteins that are going to be involved in nucleotide excision repair all start with UVR, and basically they're called UVR proteins because they're involved in ultraviolet light repair of thymine dimers. So the UVR refers to the fact that they're involved in repairing damage caused by UV light. Okay, so there's going to be different UVR proteins that are involved in this process. So it's going to start off with UVRA and UVRB here, moving along the DNA, and that complex is looking for DNA damage. And then if it finds DNA damage, then what happens is UVRA proteins are released from the complex, and then UVRC comes in and binds to UVRB, and UVRC is then, after it binds to UVRB, is going to cut this strand of DNA that contains the thymine dimer on either end of the thymine dimer. So it cuts here, and then it also cuts here. And then what happens is UVRC is going to be released or on binds. UVRD, which is a helicase, as it says on the slide here, binds to UVRB. UVRB leaves, and UVRD removes this segment of DNA, that strand of DNA that contains the thymine dimer. And then you would have a DNA polymerase come in and fill in the gap. And a DNA ligase come in and connect the two fragments of DNA together. Okay, so that's essentially what's going to happen in nucleotide excision repair in E. coli. Okay, do you guys have any questions about nucleotide excision repair in E. coli? Okay, let's see what time it is. Okay, so let's just, this is just, again, not something I'm going to ask you on a exam or quiz, but I think it's just, well, diseases aren't neat, but these are examples of inherited diseases where the defect is actually in some gene that's involved in DNA repair. So basically, if you have a mutation in a DNA repair pathway, that can lead to different types of syndromes within humans. And this is just giving you examples of syndromes where the defect is somehow in their ability to repair DNA. And that's actually how a lot of the DNA repair pathways were discovered. They were discovered by looking at bacteria that had a higher mutation rate than normal bacteria. And that makes sense because the reason they had a higher mutation rate is because they're not fixing the DNA damage like a normal bacteria would. Okay, do you guys have any questions about this topic? Okay, let's take a 5-minute break now, and then when we come back, we'll do the lecture questions for this topic, and then move on to a new topic.